Well, hello again. Today I want to talk about drug resistance. And, we touched on this a little bit in the past, but today we'll focus on it. And just want to say a few words about drug metabolism before we get into some mechanisms of resistance. Um, and there are a number of enzymes uh, found in humans as well as other organisms that metabolize natural products that they come in contact with. And so those enzymes are also used to metabolize drugs that enter into the cells. And modifying a drug can have a number of effects. It can actually increase the activity, and so some drugs are only active after they've been modified once they get inside the cell. And a very simple example that we've already seen is with the fluoro compounds, the fluoropyrimidines that inhibit thymidylate synthetase, and those are administered as just the base, but once they get inside the cell, they get made into the monophosphate, the nucleotide monophosphate, and that's the active form. But if you have a phosphate on there, they won't be able to cross the cell membrane, so they can readily diffuse in as just the base, and then they can be modified. Of course, the idea often is to decrease the activity, and that happens when they're metabolized. Um, and then sometimes metabolic changes can actually change the activity of the drug. And oftentimes there are several pathways to metabolize these compounds. So a number of enzymes that are involved in modifying them in different ways. Um, and once again, when we're talking about metabolism, the first place we should think of is the liver, and that's the prime site for drug metabolism, although there is some going on in other tissues as well. The other thing to keep in mind too is that the activity of drugs can change by interacting with other drugs that are administered. A lot of times, patients get into these cascades of drugs where one drug is administered and has some side effects, so another drug is given to alleviate those side effects, and that causes another problem, which may lead to taking an additional drug and so on. Now, metabolism in the liver, or anywhere else for that matter, generally involves making it more hydrophilic. So basically putting hydroxyl groups or carbonyls on the molecule, and that makes it more readily excreted in the kidneys, and that's the general idea. And again, some drugs are inactive in the form that they're administered in, and are converted to the active form as a result of processing. Now, when we talk about resistance and whether that's a pathogen resistant to a drug, or um, cancer cells resistant to treatment, um, we have to keep in mind this basic concept, and that is that the, the drug does not cause the resistance. It selects for, in this example shown with bacteria, it selects for bacteria that has randomly developed the resistance to the drug. So if you have a large population, again in this case of bacteria, and they're constantly changing over time, um, a rare random event may um, confer resistance to a drug particularly an antibiotic. And then if uh, that bacterial infection is treated with that antibiotic, it kills off all the ones that are sensitive, leaving the one that's mutated into a resistant form there to grow up and take over. Okay, so this is something very important to remember. These drugs are not causing resistance or causing changes in the cells that make them resistant. They are selecting for those cells that have developed a resistance due to random change. And that's the same for cancer treatment as well, and that a lot of tumors develop resistant to certain cancer drugs, and same thing's going on there. Um, there's constant change over time, especially when cells are rapidly growing, and so they can acquire resistance through a random change in the genome, and then those cells then are able to overgrow the ones that are sensitive to the drug. Now, there are a number of different mechanisms for drug resistance, and this shows some of them in this schematic of a cell. And up at the top, you can see there are these pumps that pump unwanted compounds out of the cell. And this was originally very interesting because when cells developed a resistance to one drug based on the overproduction of these pumps, so there were more of them on the cell surface, they conferred resistance to a number of other drugs as well. That was a bit of a puzzle for a while until people figured out the mechanism and realized that these pumps can pump out a number of different compounds out of the cell. And again, same thing applies to cancer cells. We have cells that develop resistance to drugs because they are able to pump them out of the cell. Over to the right in this figure now is a decreased influx into the cell. So cells can change their ability to keep the drug out rather than let it come in and then pump it back out. Probably not as doesn't happen as often as the efflux pumps, but it does. Now below that, we can get a change in the enzymes that are detoxifying the drug. We can. Now this is actually an example of a cancer cell, and so you see that we can have activation of DNA repair, and one of the targets of cancer treatment has always been to modify the DNA so that it can't be replicated. And so if you increase the amount of DNA repair enzymes, it can compensate for that. And there are some antibiotics that do similar things that, again, can become resistant by the same mechanism. Okay, as we probably all are aware, there's been a really increase in antibiotic resistance of bacteria over the last several decades, and it's been some people have suggested that we're heading into an era where it'll be very difficult to find antibiotics to treat these infections. And now, if you use a lot of antibiotics or overuse it, as the case may be, you can really develop resistance very rapidly. And it was hoped that you could do the opposite if you remove that antibiotic from from use for a period of time that you would lose that resistance. And there have been some experiments to try to do that, and they have not been effective. So it appears that once bacteria become resistant, they don't lose it very rapidly. In the lab, they can lose it pretty rapidly because when you're growing cells exponentially in the lab, which is not generally the case in the wild, because they're rarely in ideal conditions. It doesn't take much of a difference for one cell to overgrow the rest of the population. So only a minor change in the amount of DNA in the cell can have that effect and give that cell with just a little bit less DNA an advantage to overgrow everything else. But in the wild, it doesn't seem to happen very rapidly. This chart shows some of the amount of resistance to different antibiotics that have occurred over several decades. And of course, the one that gets a lot of attention in the news is this methicillin-resistant Staphylococcus, which is approaching, almost all staph is resistant to methicillin. Now, there are a number of different mechanisms for taking up DNA, foreign DNA, and these are shown here. And I might point out, I haven't seen a copy of the national boards in many years because they don't allow us to go look at it anymore. But all the copies that I have seen have had a question about this. So I can't guarantee it's still there. Maybe it's been on there so long that it's removed now. But actually one board exam actually had 2 questions about this. Anyway, this shows the different mechanisms. One at the top, transformation, and that's the uptake of naked DNA by a bacteria. And bacteria have to be what we call competent in order to do that. And some bacteria are naturally competent. Some can be made competent. Some have some signals when they become competent. Some of the streps are very competent. In the lab, you use E. coli a lot to transfer DNA around, and we can make those competent by putting them in solution of calcium for a short time, very short time, because if you leave them in there very long, it kills them. But what it does is it makes them permeable to DNA and allows the DNA to get into the cell. Now, once it's into the cell, there are a number of fates. It can exist as an extra piece of DNA, extra chromosomal piece of DNA. It can get incorporated into the genome of the bacteria, or it can be degraded. And we've seen some examples of enzymes that the bacteria can express in order to try to minimize the amount of foreign DNA that gets established in the cell, both endo and exonucleases. It can also be ligated together if there's a lot of it to form bigger pieces. Okay, so that's transformation, the uptake of naked DNA by competent cells. Transduction, part B here, is the acquisition of DNA through infection of a virus or bacteriophage. And so again, we've seen an example of a phage coming in and injecting its DNA into a cell, and that DNA get incorporated into the genome, and then under certain conditions, it can jump out of that genome, repackage itself into a viral particle, and go infect other cells. Most of the time when it does that, it does it pretty precisely, and it just excises the viral DNA and packages that. But once in a while, it can take some of the host chromosome with it. And if that happens and it goes and infects another cell, it can introduce that DNA from one bacteria into another. The third mechanism is conjugation, and that's the direct transfer of DNA from one cell into another. So they actually form a pilus, the structure between the two cells that can be used to push the DNA out of one cell and into the other. And so the cells in this case have to be compatible. And we used to think that it was only there was very little compatibility, that only a species of bacteria can only transfer that DNA to another example of the same species. But we've learned that many species now can conjugate with other species. So again, that's a mechanism that's pretty prevalent. So some species of bacteria, the main source of exogenous DNA is through transformation, others through conjugation. So it depends on the bacteria. Okay, so back to the idea of resistance mechanisms. And again, this schematic shows a number of possibilities, starting at the top with a decrease in permeability. Also, we can have inside the cell alteration of the target site, and some antibiotics inhibit translation by binding to proteins in the ribosome, and you can have some alterations in those proteins that then no longer allow the antibiotic to bind to it. And there are other examples of alterations as well. We can also find examples where the enzyme inactivation, in other words, enzyme that destroys the antibiotic. And again, these pumps that pump it out. This is another schematic of that. It has some examples on it. Starting at the top, here's some of the aminoglycosides like gentamicin and streptomycin can be inhibited from entering the cell, and this is labeled here as a low-level resistance. So you could overcome that with higher doses, but there are also enzymes inside the cell that modify the antibiotic and destroy it, or alter their examples, as I mentioned before, altering the target site. And again, this would be for these antibiotics, be altering the ribosomal protein that it interacts with, and that generally gives a more high level of resistance. Let's see what else we have here. And of course, down at the bottom, we again see an example of increased efflux out of the cell by by increasing the number of these pumps that pump the molecule out of the cell. We also see over on the right-hand side reduced binding affinity, and we'll talk about the lactams, ampicillin being the most common example of that in a second. And down at the bottom, an example of vancomycin, which inhibits the same pathway as ampicillin's reduced binding affinity. So one of the major forms of resistant to penicillin is the enzymatic inactivation of the drug, and this shows uh, how that works. And you can see penicillin up here on the left, and it breaks the four-membered ring here, which inactivates the antibiotic. Now, one of the ways that we've gotten around this is we have an inhibitor of that enzyme. So often it's administered along with penicillin to inhibit the inactivation of the enzyme, and that's marketed as a drug, Augmentin. So Augmentin is penicillin along with an inhibitor of this beta-lactamase, the enzyme that inactivates it. Okay, so again, an example of converting a drug-sensitive cancer cell into a resistant cell. On the left, we have the drug coming into the cell, interacting with the target, in this case a protein, and resulting in the death of that cell. A lot of cancer drugs, now there are many more cancer drugs available these days that target specific types of tumors and are more directed towards the tumor cell, but a lot of the drugs are basically directed towards fast-growing cells, so that they will inhibit the growth of any cell. But since the cancer cells are growing faster, they tend to be affected more. They tend to take up the drug more readily and to incorporate it into the mechanism that will kill the cell. This is the same, so I should say similar approach used with weed killers on your lawn that target these broadleaf plants, and basically they're targeting tissue that's growing very rapidly. Okay, on the right here, if we have now a mutation in the target, the gene for the target, and again, this is a randomly occurring mutation, and now we've just selected for that by treating it with the drug and killing all the ones that are sensitive. And now the drug cannot interact with the target, and so the target is still active, and so the cell survives. Okay, so this now puts some of these mechanisms in a list. Again, that decrease in influx in the cell can make it resistant to a drug, and the increase in efflux can also have the same effect. And these pumps that facilitate that generally will remove a number of different compounds or drugs, so that if you develop resistance to one drug, you may develop resistance to other drugs as well. Also, the induction of these detoxifying mechanisms can affect resistance as well. Now, we can have reduced drug uptake due to receptor regulation. So by modulating the number of receptors on the cell surface, we can increase the amount of uptake of the drug or decrease it. And there are a number of receptors that regulate the amount of uptake of the drug, and we don't need to go into them, but here's the list of many of them. Okay, so detoxifying drugs can happen in two ways, in two phases. First is the addition of a polar group, so the oxidation or hydroxylation, as I mentioned before, to make it more water soluble or hydrophilic. And this is carried out by this family of proteins that contain the cytochrome P450. I think you've talked about cytochromes a little bit with Dr. Bartling and electron transport system, and these are the same type of molecules, these complex ring structures that were used there, and they're used in this case to modify compounds and make them more water soluble. Also, we can get conjugation where different functional groups are added. Again, these are oftentimes very soluble molecules that make it more easily, make the drug more easily secreted through the kidneys. Now, this is a complicated drawing. I just want to point out that, again, there are a number of these P450 molecules that carry out different types of, generally some form of oxidation. Okay, just a few things about cytochromes. Again, these are heme-containing proteins, and they react with cobalt. And, you know, there's a large number of them. I'm not sure how many, but they are divided into different families that are similar to each other and have different functions. Some examples are shown here. Not really necessary that we go into that, but there are a number of alleles of each locus, so there's a lot of variation from one person to another, and that's why some people can metabolize some drugs faster than other people. And this is an example of some of the cytochromes and some of the different compounds that they will metabolize. And you can see there's quite a variety of different types of compounds. For instance, the first one, the 1A1, substrate caffeine, as you recall, is a purine analog. And of course, testosterone is a steroid. So there's quite a bit different types of molecules that this enzyme can act on. And so there are these transporters, again, that are involved in transporting the drug across the cell membrane, and these are called ABC transporters. They bind ATP, so ATP binding cassette transporters. And again, these are involved in multi-drug resistance and a lot of different cells. So these proteins generally have two domains. One that's the transmembrane domain that's involved in the actual transport, and then this nucleotide binding domain, which binds to ATP and facilitates the transport. So this is an energy requiring mechanism. So and again, there are a number of these types of transporters in humans, 48 known transporters. We can once again divide them into different families based on their sequence, amino acid sequence. And this is a list of some of those families and the differences between them, which I don't think are real important. I think the main issue is, again, that we have, our cells have these transporters that allow them to pump molecules out of the cell. They can generally uh, act on very different molecules, and we can get resistance to drugs because they can be overproduced. So if you add extra transporters to the cell surface, you can transport more of the drug out of the cell. Now, there's been an effort to try to come up with an inhibitor of these transporters to counteract the drug resistance, and so far it hasn't really yielded very good results. I'm assuming that Dr. Bell talked about RNAi and how it can be used to inhibit gene expression, and that's another potential approach to dealing with the overexpression of these transporters. And this is a real problem because you could develop resistance. If you develop resistance to a drug because of the overexpression of the transporter, it may confer resistance to other drugs as well. Now, another mechanism, particularly in cancer treatment, is to, again, try to damage the DNA. And so there are a number of drugs that do this. Many of them are alkylating agents that derivatize the bases on the DNA chain and prevent either transcription from going through or the DNA polymerase during replication. So by overexpressing the repair mechanisms, the enzymes that repair these modified bases, we can develop resistance to these drugs as well. So, as fast as the drug is modifying the DNA, if the cells can repair it, then there's no damage done. And we've talked about a number of different types of repair mechanisms, and there's a lot of redundancy in DNA repair processes, so that different mechanisms could be used to repair the same type of lesions. There's actually a bacteria that can repair damage due to X-ray exposure, and as you remember, X-rays cause double-stranded breaks in the DNA, and so you can kill cells with X-rays by breaking up the DNA. But this particular bacteria can repair an amazing amount of that damage. So you can break the DNA up into little pieces, and the bacteria can put them all back together and make a viable chromosome. Okay, so I'm not going to go into the details of the different types of repair mechanisms since we've already talked about those, really. And this is just some more examples of the repair process, and again, we've been through this. Now, one thing I do want to mention is that this is a complex of proteins that carry out different functions. First function, of course, is to recognize the damage, and then to remove it. Then second thing is to remove it, and then third is to fill in with new DNA. And again, that requires all these enzymes, polymerase. It doesn't require primase because you already have something to elongate off of, but it requires the polymerase and the ligase to seal the two pieces of DNA together. as well as all the other enzymes that are needed for DNA replication. And this is a list of some of those proteins and what they appear to be doing. Of course, we don't know the function of some of them, but many of them we know. And again, there's nucleases involved, and to cut out the bad sequence, the polymerase helicases to keep the two strands apart, and then proteins that stabilize the whole region and bind to single-stranded DNA to keep it apart during the synthesis of the new strand. And the base excision repair is just one example of a type of repair where one base is removed and basically a new one is put in its place. And that's shown in the schematic on this picture. Again, this is just one type of repair, and there are many. And again, if you have a lesion in the DNA, it could be repaired by any of a number of different mechanisms. Although there is some specificity, so base excision repair will generally repair these kind of oxidative lesions and a variety of other lesions. And so I just want to mention again this idea of mismatch repair that we talked about a while back, and that is if there's the two bases opposite each other on the complementary strands are not complements, and so one of them is wrong, and so it has to be removed and replaced with the right one. And again, the cell needs to try to figure out which one's the right one and which one's the wrong one. And in newly synthesized DNA, it can tell that by methylation patterns, where the old strand is methylated, the new strand is maybe not yet methylated, and so in that case, the repair mechanism will repair the new strand to be a complement to the older strand. Now, if you get a double-stranded break in the DNA, this can be repaired, and again, by a homologous recombination event where you can use the homologous chromosome as a template to synthesize new DNA on the other chromosome. In this case, we have a break in the red chromosome at the top, loss of some DNA there, but then that end of the red chromosome can invade the blue chromosome, displacing the other strand, and then be used as a primer to synthesize new DNA. And then this can be resolved in a couple of different ways, one that causes a recombination event and one that doesn't, and you can look at those at the bottom and figure out why that might be the case. Now, the last thing I want to talk about is something I mentioned earlier a while back, and that's the idea of gene amplification that causes drug resistance. And the first known eukaryotic example of that came from a cancer cell that was, or cancer line that had been taken from a tumor that was resistant to this drug methotrexate. And that methotrexate is an analog of folic acid, and so it targets the enzyme dihydrofolate reductase, which is required for thymidine synthesis, as you remember when we talked about nucleotide metabolism. Now, you can get cells that are resistant to methotrexate by a number of different mechanisms. One is alteration of the enzyme that then doesn't bind to the drug, and there are examples of that. You can also get, the pumps that pump it out of the cell that confer resistance. But also you can get increased copies of the gene that then make more RNA, which makes more protein. And so basically just overwhelming the drug. And particularly if that happens in a tumor, you can only treat patients with so much methotrexate, or or it'll be toxic. So you can't increase the concentration all that much to overcome the resistance. But in the lab, you can take these resistant cells and you can increase the drug concentration and select out cells that have even more copies of the gene, and then you can take those and increase the concentration, and the stepwise selection of increasing the dose of the drug, you can isolate cells that have more and more copies of the gene. And so there are cell lines that have as many as 1,000 copies of the dihydrofolate reductase gene. There's been other examples of gene amplification as well. And in fact, a drug that inhibits the adenosine deaminase gene can be used to amplify that up many hundred or many thousands fold. And so these cells then carry this extra DNA with them in one of two forms, as we talked about before. Shown here on the left is this extra chromosomal DNA that has been called a double minute chromosomes, because when they condense here during mitosis, you can see the two homologous pieces of DNA still paired to each other. Now these aren't going to bind to the spindle and assort perfectly like normal chromosomes that have a centromere. So they get distributed in the daughter cells randomly. And in this case, if you take the drug away, you very rapidly lose the resistance, because again, those cells that have not inherited as much of the double minutes don't have as much DNA and are able to replicate their DNA faster and divide faster, and so will quickly outgrow those that have more copies. Now on the right, we see some cells take that extra DNA and incorporate it into a chromosome, and that's a big yellow stripe or band in this in the chromosome towards the bottom of the figure, which has been sometimes called a homogeneously staining region, because it doesn't show the banding patterns that chromosomes show when they're stained with these chemicals like Giemsa stain. Now this is much more stable, and so removing the drug from cells that have an HSR generally does not, these cells don't lose resistance to methotrexate at least over short periods of time, not like the double minutes do. So this again is a somewhat common form of resistance, because at any given time, as we saw when we talked about genome structure, part of the genome has been duplicated. And so again, we're just selecting for those cells that have duplicated, in this case, the dihydrofolate reductase gene. And we can see this in bacteria as well. Okay, we're going to stop there today and move on to another topic next time.